Hello, I'm Shalev Vitskovich from the Weizmann Institute. And on behalf of Dr. Arjun Raj and myself, I would like to welcome you to the 2021 Keystone Symposium on single-cell biology. This week, we won't be doing any skiing or hiking around the Ruiz, but we do expect 3 days of really exciting science ahead of us. I'd like to use this opportunity to thank all of the Keystone Symposium staff for making this virtual conference happen. I'd also like to thank all of our speakers for their willingness to participate and for their flexibility in coping with the challenging time zones. And of course, you, the participants, we have more than 600 registrants from 43 countries. Throughout this meeting, there will be several opportunities to interact, which we highly encourage. There's going to be a Q&A session after each presentation, and you can post your questions and vote on other people's questions. We can use the poster booths to interact with the poster presenters during the poster session. We're going to have one-on-one connect sessions where you can request an appointment with speakers and journal editors to discuss scientific career development and publishing questions, and also 2 exciting roundtable events. And you can find more information on these and other networking opportunities in the meeting homepage. So I'm going to turn it now to Arjun to introduce our keynote speaker.
Thank you, Shalev, and thank you all for attending. I think we have a really wonderful meeting in store. It is truly an honor and a pleasure to introduce Michael Elowitz, Professor of Biology, Bioengineering, and Applied Physics at Caltech and an investigator of the Howard Hughes Medical Institute, and to introduce him as our keynote speaker to kick off the Single Cell Symposium. Michael is, of course, highly accomplished, has won many awards, and I won't list them all here in the interest of time because it's more fun to hear his science. I'd like to highlight maybe a couple of things that I think are really particular to Michael's work and how I think in many ways point to the future of our field. In many ways, our field has been dominated by technical innovations, both from the advent of GFP and imaging, the single molecule imaging, and all the current proliferation of sequencing-based methods. Throughout all this, Michael's work has really distinguished itself by its conceptual novelty and elegance that provides these just penetrating and often very orthogonal insights into the underlying biology. Perhaps the best way of putting it is that, some work makes you think like, how did you do that? And Michael's work always makes you think, why didn't I think of that? It's just so beautiful. Some examples that really stand out to me, so much so that I taught them in my graduate course, are his beautiful work on intrinsic and extrinsic noise from 2002, which in many ways ignited this field that we're all interested in right now. And another was actually his beautiful work on frequency modulation with a long chi from 2008, which just completely pointed to a new way of thinking about how genes were regulated in a quantitative manner. These are just 2 examples from a very long list of papers that I really think should be required reading for our field. So, at the risk of mildly embarrassing Michael, I actually very much remember the very first time I met him. I was out at Caltech, and I remember we hung out and he got me a smoothie. It was really nice. And I remember at the end, I said, I just have to tell you, your work is what inspired me to get into this field. And I still feel that way to this day. And I'm sure his talk today will provide similar inspiration, hopefully, to all of you. So with that, take it away, Michael.
Okay, well, thanks, Arjun, for that really nice introduction. I always felt you were a kind of kindred spirit. And in fact, we even worked on the same system and competence in Bacillus back in the day. And thanks also, Shalev. So thanks to both of you for organizing this amazing meeting. I think everybody here would agree there's a kind of revolution going on in single cell biology, which is why this meeting is happening. And so I wanted to, what I really want to tell you about today is kind of one project we've been working on to try to think about how we could create synthetic circuits, kind of take a synthetic biology approach to understand how cells can exist in such a huge variety of different states in a controllable way as we see them in real organisms. But to do that by, again, synthetically. So I thought I would just start by just reminding myself and you that the idea that cells are incredibly diverse is something we've known for well over a century. It's always best illustrated in these beautiful drawings by Ramon Cajal. This one is one of the retina. But we know that we've always known that cells exist as huge variety of different shapes, sizes, morphologies, and also now molecular states, right? So if we fast forward to a few years ago, we've been able to see that with single-cell RNA-seq, just this huge variety of different molecular states, different RNA transcriptional profiles, different chromatin states, and so on. And so we're really getting a deeper and deeper picture of what the variation looks like among different cell types in an organism. And so we can see that here molecularly, and with spatial transcriptomics techniques that Shalev and Arjun are great pioneers of, you can also see not only what those states are, but where they are in space, how they're related to each other spatially, and what kind of larger scale structure you have. So this example is from one of Long Kai's papers on seqfish. And we're even going further. So it's not just what the cell types are and where they are, but also kind of how they're related to each other. So there are this sort of set of approaches that we and others have been working on to try to reconstruct lineage relationships among cells by engineering barcodes that can be dynamically edited so that you can reconstruct the relationships among cells, much as you would reconstruct the phylogenetic relationships among species by looking at the similarity of stochastic edits in barcodes. So in this way, we're getting kind of a glimpse into the past of cells and into the relationships among cells, not only their current states. So we can observe all of these features of states, and we can also, I think, in a kind of parallel set of parallel research thrust over the years from regenerative medicine, we've also learned how to steer cells into many of these states using inhibitors or ligands for different signaling pathways. So I love this map, which just shows how you can produce this incredible variety of different cell types, starting from pluripotent cells by sequentially activating or inhibiting different signaling pathways, or in some cases, expressing different transcription factors in the cell. So in one point of view, then, I think we really have an amazingly broad and deep look at what cell states are like and how we can manipulate cells kind of operationally. And this is all great. But on the other hand, if you try to ask the question of, how do the underlying circuits establish all of this incredible variety of fates, and how do they establish those fates in a way that cells are able to kind of navigate from one state to another in a very controlled and constrained way, I think that question has been a little bit harder to deal with. Of course, we know a lot about the different, again, signals and transcription factors that are controlling cell fates, but when we try to map them as circuits, they often appear really complex. Here's just one example. I could pick kind of any of these. There's a lot of different transcription factors involved and a lot of different regulatory interactions. And it's hard to understand by looking at these diagrams what subset of these interactions or what aspects of this circuit are really sufficient to give you this key property of multistability and controlled transitions, okay? So this is really the theme of this talk is how can we get insight into that question? What are the circuit design principles? What are the architectures that are sufficient to give you this incredible range of cell fates and transitions that we see in nature? And to address that question, we wanted to kind of take a different approach, kind of the opposite approach of trying to analyze the natural system, and that is asking whether we could create a synthetic system that recapitulated some approximation of this behavior, of the ability to generate lots of different states. So the overall idea here is that we would take a synthetic circuit, engineer it into a cell, and try to design the circuit in such a way that it could allow the cell to exist in many different states. Okay. And the ideology here is something that we really like in the lab, which is that there's an interplay between natural circuit design and synthetic circuit design, where synthetic circuit designs can often take advantage of principles that we learn from natural circuits. And at the same time, they provoke new questions about natural circuit design that we might not have thought to ask. And I hope that that's the case in what I'll tell you about. So this idea of trying to build a synthetic cell fate control system it was something that we started talking about a number of years ago with an absolutely brilliant graduate student. And I say that in the strongest possible terms. So Ron Zhu has kind of developed this project that I'll tell you about from scratch, and is just brilliant kind of in every dimension, from computational, theoretical, and experimental. And the starting point was we asked, okay, what features would you want to have in an ideal cell-fate control system? Well, the first thing is it has to, of course, produce multiple states. So you can think of those as just different colored cells, or you can think of them as attractors in the space of transcription factor concentrations. So that's what's illustrated here with these 3 points. Each of them is stable. So these are points in the expression space of 2 transcription factors. This is just schematic, of course. And we're imagining that if we push the cell up, we added a little B, it'll come right back. These are stable fixed points. So that's one feature you have to have, just multistability. But it's not only that. We also don't want these to be perfectly stable. We want to be able to switch cells from one state to another in a controlled way. That's another essential feature. And if we start to push a little deeper, we see that real cell-fate control systems have other really interesting features beyond just multiple states and the ability to switch. They also undergo bifurcations or irreversible differentiation events, where you could have, let's say, a pluripotent cell that you could destabilize, but without destabilizing to, let's say, more differentiated fates. So the question is, can we also reproduce that feature of kind of irreversible fate commitment events? And finally, real systems have not only the 3 states that I diagrammed here, they have many states. And so an ideal architecture would be expandable. So would it be able to allow us to grow the system to have more and more states? And that would also recapitulate something that happens during developmental evolution, which is the growth in the number of different states that you have as organisms get ever more complex. So both from a usefulness point of view and also from a way of understanding evolution, it would be good if the system had this property of expandability. Okay, so how are we going to do that? What kind of design would actually be useful for making a synthetic multi-stable system? And here's where Ron went to the literature and tried to kind of read a lot of papers about the circuits that control natural fate, cell-fate systems. And these are just 2 examples, one from the classic case of myogenesis, another from pluripotency, equally classic now. And we noticed a couple of themes, okay? One is that there's often sets of transcription factors that operate as homo and heterodimers. So they don't just work alone as a monomer, they work as either a homodimer or a heterodimer. Those homo and heterodimers can act as activators or inhibitors in different cases. So that's one theme, is the use of dimerization. And the second common theme in many of these systems is a lot of positive feedback, where these transcription factors acting as homo or heterodimers autoregulate their own expression in different ways, directly or indirectly. Now that makes sense. You really want to have positive feedback in there in order to stabilize different states. But what Ron did is he took these 2 themes, kind of promiscuous multimerization or dimerization and autoregulation, and came up with a circuit design that really crystallizes just those 2 themes in the simplest possible way. And I'm going to show you that design now, and the rest of the talk will really be about this design. So this is the simplest version of it. It's MultiFate 2. So we're starting with 2 instead of 1 because 2 means 2 transcription factors. And later I'll show you 3 with 3 transcription factors. And in this design, let me go through it real quick here. So you have 2 transcription factors. Let's call them A and B. We're going to imagine A can homodimerize to make an AA homodimer, and that AA homodimer can activate expression of A itself. So it's a positive autoregulatory feedback loop. Critically, the A monomers do not activate. They don't do anything. Okay, it has to be dimeric in order to activate. B works exactly the same way. B forms B homodimers that activate B. And then critically, A and B can form a heterodimer that's inactive. It just doesn't do anything. So in this design, you have both positive autoregulation of each transcription factor and cross inhibition at the protein level. Now, one really nice thing about this system is it's simple enough that we can analytically describe it with a relatively small number of equations. I'm not going to go through them here, but I just wanted to show you how relatively simple the system is. There's a set of equations that just express the idea that these different proteins can competitively homo and heterodimerize with each other in equilibrium. And then there's a set of differential equations that just describe the evolution of the total concentrations of A and B and the activation by the homodimers of A or B. Now, when you analyze those equations, you can write down or draw this sort of phase diagram. And on this diagram, again, this is the concentration of A and the concentration of B. These three black dots represent stable fixed points. So for these values of the parameters, which are reasonable, you can get what we call, we call type true tristability, but the point here is just it's tristable. It has three states. These white dots are unstable fixed points, and the colored regions are the basins of attraction. So if you start the cell at some point in space, you can see where it will go to. Okay. So just to make this absolutely clear, if you're here at this stable fixed point, it means you have an excess of these B homodimers. Here you have a lot of the A homodimers, and here in the middle you have a mixture of A and B. So three states in this regime. Of course, this is not the only regime. There's actually a range of different behaviors you can get. And by varying parameters, you can switch the cells from one phase diagram to another. So for example, I'll show you later, we can destabilize by reducing protein stability of the transcription factors. You could destabilize this fixed point and go from a tristable to a bistable regime. Okay, so the dynamics are quite rich, even though the circuit's really simple. Now, what about that property of expandability? This is where the circuit is really nice, because if we want to, we can plug in a third transcription factor to an existing circuit. So if I add C, as long as C can heterodimerize with A and B in an equal way, it can participate in generating these inactive complexes, and all of these transcription factors can mutually inhibit each other at the protein level. Now, that at the protein level part is nice because that's what makes this expandable. If we took a conventional transcriptional toggle switch design where the two transcription factors transcriptionally repress each other, and then we wanted to add a third one, we would have to then go back and clone in binding sites in the first two so that the third one could repress them. So you would have to change the existing circuit in order to expand it. So that's not true with the MultiFate design. Okay, so now we're going to go from kind of the idea of the circuit to how do we actually build and characterize it, and what does it actually do? So if you think about it, the design features that we need are relatively simple. We need transcription factors that activate only when they're homodimeric. They have to be able to inhibit each other through heterodimerization. And there's a third property that I didn't really touch on yet, but is practically important, which is we would like these factors to be as externally controllable with inducers of small molecules, for example, as possible, so we can manipulate the system. Okay, so the basis for creating this is these amazing proteins, the zinc finger DNA binding proteins, and many groups have worked on these over the years. Mo Khalil's been doing some really beautiful work with them recently in particular. And so Ron took some of these characterized zinc finger DNA binding proteins, and he engineered them to have an activation domain and a pair of, and have them bind to binding sites on a kind of reporter construct. And the idea here is that the individual binding site, the individual zinc finger protein recognizes a 9 base pair site, and that interaction is too weak for a monomer to activate. So when you express the monomer, you don't want to get any activation. But if you attach a leucine zipper from the protein GCN4, then these proteins can homodimerize, and those homodimerize can bind efficiently and activate the target gene. So in this way, if you look at the activity of the homodimers versus the monomers, you can see the homodimers are much more active. But you still see there is some residual activation by the monomers. And Ron was annoyed by that. And so he added 2 arginine to alanine mutations in the DNA binding domain of the zinc finger that pushed down this background activation by the monomers even further while preserving most of the activity of the homodimers. So in this way, we get homodimer-dependent activation. Okay, the next thing we wanted to have is kind of external control. So we replaced that first homodimerization domain I showed you with FKBP, which is a very well-known protein binding domain that can be controlled by the drug AP1903 through chemically induced dimerization. And we also attached these little black bullseyes, which are degrons, which kind of destabilize the proteins, but can be inhibited by a drug called trimethoprim. And in that way, we can control from the outside the stability of these proteins. We also attached fluorescent protein reporters. And so when we do this, now we have controllability, where you can see here on this plot that these transcription factors can positively autoregulate their own expression, turn themselves on, only when you have both drugs, the AP1903 and the trimethoprim. In other words, when you stabilize the protein and allow it to homodimerize. Okay, the last properties we need the heterodimerization to kind of allow inhibition. And so here's just an expression experiment to show how that works. So imagine I take one of these engineered transcription factors, I let it positively autoregulate, but I transiently transfect in a competing transcription factor that shares the same DNA binding domain. So if I do that, that knocks down activation by the first protein. And of course, it doesn't do that if I use a different dimerization domain or no dimerization domain at all. So we see that in general, heterodimerization can block or inhibit activation, and we can do that in both directions. Okay, so with all of that out of the way, we can now design the actual circuit, which has, in the case of MultiFate 2, these two transcription factors, each with its own fluorescent protein. We have different versions of these circuits with different numbers of binding sites, and then we integrate them by piggyback into the genome. And so and we isolate clones. And so each clone will have kind of a different configuration, different relative copy numbers of these two different genes. And we do this I should say, in CHO-K1 cells. Okay. So now, does it work? This trouble with synthetic biology is you can do it, and then it just might not work. But okay, here, I probably wouldn't be telling you about it if it didn't work. So here's what happens. So if we start off with the system off, with no drugs at all the cells are at low levels of both transcription factors, as shown in flow cytometry. If we then add the two drugs together, wait 3 days, we see that all of these off-off cells have now gone to one of three possible activated states. So that you see the tristability that we predict here in the model, where you get either one protein, the other protein, or both proteins at elevated expression levels. So this is really showing you that this state is fundamentally unstable when you add the drugs, but the system is at least trimodal, and I'll show you in a second that it's also tristable. Okay, so that's shown us, that we have these three states, but are they really stable states? Okay, or are they switching back and forth? So to look at that, we fax sorted each of these, the cells in each of these three modes into one of three wells, and then we cultured them out to 18 days. And then you can see that each of these states is really stable for quite a long time. And this 18 days is just the point at which Ron decided to stop doing this experiment, but they're really long-term stable. And in the next slides, what I'll try and do is I'll try to simplify these plots into these kind of quadrant plots. So these are just 4 circles representing the bottom left, just one color, the other color, both colors, and no colors here. These 4 circles, and the color is representing the fraction of cells in that state, just to kind of simplify the presentation. I hope that's clear. So these guys are like these guys, and so on. Okay, but instead of flow, it's actually more satisfying to see it as a movie. So here are those 3 states, which are we're just seeded individual cells that are started in either the A state, the B state, or the A plus B state, again, identified by these 2 fluorescent protein colors and 3 color combinations. And what you can see in this movie is that each colony retains, as it grows, the state of the first cell that seeded it. So this is just a way to visually see the stability of these 3 states. So this is what tristability looks like in the microscope. You can also zoom in on individual colonies to kind of see this. These are really large-scale movies that are made by tiling an array, tiling across the surface of a plate. Okay, so that shows that we have these states and they're stable, but can you actually switch them? That was kind of our second requirement. So one of the things that I didn't mention before is that Ron really engineered an additional level of control, which is inducibility with additional binding sites in the promoters that you can use to induce ectopically expression of either of the 2 transcription factors. So if we start off in this state, which is the A-only state, where mainly A is expressed, then we add DOX that ectopically expresses additional B. You can start to see this state depopulating, and cells going to this state and this state, and then 3 days later, you see most of the cells here in this B-only state. And then if we remove the DOX, you can see they stay stably in that B-only state. So we've kind of switched them from the A-only state to the B-only state with a transient pulse of DOX. You can also go the other direction with a different inducer, 4-OHT, tamoxifen, and there you see kind of the opposite behavior. You depopulate the B-only state by adding A. You go through this intermediate high state and come over here to the A-only state, and then you remain stable there if you wash out that inducer. So these states are switchable as well as stable. Okay, now let's think about some other properties. So a lot of these fate control systems have this amazing property of irreversible differentiation. And I think nobody has done a more beautiful job of explaining this property of irreversibility than Jim Farrell, who had studied it in particular in the system of Xenopus oocyte maturation, where addition of progesterone transiently induces a transition from a kind of immature to a mature oocyte that can then be fertilized and grow into an embryo. And this transition is irreversible. You can't go backwards. And we see similar things, of course, in lots of cell fate, lots of differentiation systems where, for example, you can exit from a pluripotent state to a more committed progenitor, but then the cells don't go back the other way. You also see cases where they do go back and forth, but in general, there's a lot of cases where there are these irreversible events. Now, one nice thing about this circuit is it actually provides a natural way to demonstrate that as well. So what I'm showing in this animation is kind of a simulation. So the little red dots here are simulated cell trajectories. And what we're doing, let me just back this up for a second. I'm starting here in that same tri-stable state. Now I turn down the protein stability. That eliminates that third fixed point at high A and high B, but it preserves stable attractors that are just A or B only. And then if I turn the protein stability back on again, in a second, that state comes back, but the cells remain in the A only or B only state. Okay, so in this way, transiently, I can destabilize this A-B state and get the cells to go to one or the other, and then they're stuck there, even if I restabilize that state. Okay, so that's kind of a prediction for the system. And we can do that because that trimethoprim, which controls protein stability, is something we can manipulate. So if we start off with high trimethoprim in this original regime where we have the cells, we can start with cells that we've sorted out only from the high A and B state. And then we can put them in this low trimethoprim state. Then they depopulate the, that high state, and they go to either the A only or the B only state. And then they remain there stably if we put back TMP. Now, what about expandability? So when we go to 3 transcription factors, we can expand this system by adding a third zinc finger. If you do that, the phase diagram looks like this. So these colored regions are the different basins of attraction for the different states. And there's 7 of them. Okay, so now we can see, can we actually get 7 states with the system? And so if we do the same experiment, we're adding the inducers to the cells when they're off. These are cells now that contain all 3 genes, produces a range of these different states, which I've now diagrammed here with this hexagon. So the ABC state is in the middle, and the other states are indicated. Then you can see that if I look at day 0 versus day 18, for cells that were sorted into each of these 7 states, you can see that all of these are stable over long periods of time, with the exception of the off state, which is fundamentally unstable, and it goes into other states. So there's a little bit of asymmetry in this particular one where it tends to preferentially populate certain other states when you destabilize it. So the point here is that this MultiFate 3 circuit actually generates 7 states. And of course, again, this is most directly visualized now in a movie where we have 3 colors, 3 fluorescent protein colors, and the various color combinations of them. And what you're looking at here is cells that are initially started in one of these states, and you can see that each of the states, again, is really stable over long periods of time. And in fact, Ron ran some of these flow cytometry experiments. These are the movies, but he ran the flow experiments out to about over a month, and you can really see these states remain stable. Okay, so finally, the other thing that we see a lot in these natural systems is again, kind of following that theme of irreversibility, is this sort of progressive commitment to different fates. And with these 7 states, we can now see something like that going on in the synthetic system as well, where if we take that same parameter, the protein stability, and we just progressively reduce it from a highly stable proteins to intermediate to low, we progressively first get rid of the ABC state, leaving the other states. And then if you go even lower, you get rid of the pairwise states, the AB, the BC, and the AC, leaving the A only, B only, and C only. That's a prediction from the model. So we can do that experimentally. We can progressively go down. So here we see 7 stable states at high trimethoprim. If we reduce that, we see we've gotten rid of the ABC state, but we still see stability in the other states. And if we go even lower, then we've destabilized all of the states except the A only, B only, and C only. And so now we've kind of progressively reduced the degree of multistability. in the system. So it kind of recapitulates something like that kind of progressive commitment. So what about the scaling? So I showed you from 2 to 3. Can we go beyond 3? So in the model, if you analyze the circuit, it has this really beautiful property that the number of states scales exponentially with the number of transcription factors. So if you imagine going out to, say, 10 transcription factors, you're approaching 1,000 different states. And also the basins of attraction of those states are relatively balanced, so they're relatively equally accessible. So we think that this system has a beautiful scaling property. Okay, so what I hopefully showed you is kind of, what we're thinking about this is kind of one step towards a kind of dream of a synthetic multicellular organism. This is kind of the core fate control system. And of course, we will need to also have communication and population control. And in other projects in the lab, we've been working on these other aspects of trying to kind of build out to a more complex synthetic system, synthetic multicellular system. Okay, so just to summarize, I showed you this design, MultiFate. It's naturally inspired. We didn't just make it up. It was really came from features that we see in natural circuits, and it seems to really work very effectively. I showed you how it allows controlled switching between states and these irreversible transitions, hierarchical commitment, a lot of the features you see in natural systems. I think to me, one of the most striking things is, as a systems biologist or a synthetic biologist, I'm always amazed when circuits actually behave roughly as we expect from the model. And in this case, the model predicts not only the sort of stability diagrams, but also more subtle features such as the bifurcations. And then at the end there, I'm showing you that it kind of scales. And I think that's a really critical feature if we want to build circuits that kind of go beyond just a few states. And so for that reason, we think of it as kind of a basis for future work on synthetic multicellularity. So let me just thank my amazing student. Again, Ron is just spectacular and really led this project from start to finish, every aspect of it. We also had a really wonderful summer student in Mexico working remotely, looking at different stochastic modeling aspects of the circuit. And we also had lots of wonderful discussions and interactions with Jordi Garcia-Halvo as well. So thanks very much.
Okay, thank you very much, Michael, for this amazing talk. We have quite a few questions. I think I'll start. Is there any idea to add noise in gene expression, different promoter strengths, to allow transitions without needing to add or remove doxycycline, i.e. add an internal mechanism to modify the number of stable states?
I see. So is the question, can we add noise, can we control the spontaneous transition rates by playing with the noisiness of the promoters? I think that's the question. And I think that's a beautiful question. We have not done that, but I think it's certainly something that could be done because noise is something that you can control in these synthetic promoters. In other words, you can get the same mean expression with different levels of variance. And I think from the model, we would expect it to affect the spontaneous transition rates. So I think it's a great idea. Yeah.
We have a question from Joshua Thyssen, which is, why does decreased protein stability destabilize only the heterodimer-dependent stability point and not the homodimer-dependent stability point?
Okay, so what's happening when we change the stability of the proteins, we're changing, first of all, the stability of all the multi-phase proteins together. But it just turns out that when you look at what happens, those multiple the heterodimer, sorry, they're not heterodimer states, but they're the states that involve co-expression of multiple transcription factors. Those are more sensitive to that parameter. So just as we reduce that parameter, those are the first ones to disappear. And I guess the reason I would say that is maybe to answer your question, that in order to maintain those states, you need the homodimer levels to be high enough that they positively autoregulate, despite the fact that a lot of the monomers are consumed in those heterodimerizing interactions. So I think that's why those states are more sensitive to protein stability.
Yes.
Sorry, I'll just ask my own little question here, perhaps related. So for the few cells that do leave the state, like every so often you see like a couple that'll sort of jump ship, do they move to predictable other states, or do they just kind of randomly pick a state? So do they sort of jump to a state that makes sense, like it's only one hop away?
Yeah, it's a great question. Like, So one thing I did not really get into is that we have different variants of these different cell lines that have the circuit, and some of them have biases, and sometimes strong biases, where there's kind of a preference. So you can access all the states if you push those into them, or low frequency they get populated, but some of them are really biased. And so depending on which clone it is and probably the relative expression levels of the different transcription factors in that clone, you do see these biases in different cell lines. And so we've tried to kind of select a variety that have different biases. But, yeah.
Okay, there was a great question here from Tiam Heydari. Is there any study about the change of protein stability during differentiation, sort of in natural stem cell maintained tissues? An example where protein stability changes and that causes a drift to these stable states?
I would say there are, I'm trying to think offhand of a good example. I think there are cases where, proteins are destabilized during differentiation. Yeah, I'm not sure I have right off the top of my head a good example of that. It's a good question. Yeah, so.
Joaquin Navajas Acedo has a question. Your GRN, gene regulatory network, takes 3 days to get activated. Is this a bug or a feature? So how does your system recapitulate changes in gene expression that are very fast or very slow, like circadian rhythms?
Yeah, that's a good question. So I didn't talk too much about the dynamics, but you're right that depending on what the transition is, the time scale required to switch can be long or be short, and can actually be tuned in the model in different ways. So I guess the question is kind of, the question is kind of like, is that the right time scale, I guess, to do? What's the ideal time scale to be on?
Yeah, I guess it's just kind of like, what can you do about it?
Yeah, I think it kind of depends. There's been, I think there was really nice work recently, showing that, very brief transient exposure to BMP signal could actually be sufficient to initiate an irreversible differentiation process from pluripotent state, and that initiating signal didn't have to be long, like it could be, I think, 30 minutes or an hour. But nevertheless, the subsequent changes played out over a longer timescale. So I think there is a question, which is, what is the natural timescale or the ideal timescale for these events, and can we make it faster or longer? I think there's probably some trade-off. If it's too fast, the system may be sensitive to, small variations in input. If it takes too long to integrate a signal and switch, then, maybe it's too slow for development. So yeah, I think that's something we probably will have to explore in the future.
It's actually, I mean, just as a total aside, I think it's kind of interesting to think that, we always think about stuff growing in addition on the order of hours or days, and many organisms takes months to years plus to develop. It's like such a different time scale. So anyway.
Yeah.
Okay, I think maybe I'll ask one quick last question. Can you say anything about your plans to incorporate juxtacrine signaling or cell-cell communication into these models?
Yeah, let's see. So communication is essential for multicellular development. We've been studying, I think one of the themes of our lab really is looking for weird circuit designs that have some cool properties. So one of the things we've been trying to do is look at a motif that we see naturally in a lot of signaling systems, which is the use of promiscuous interactions between many ligand and receptor variants. And we think that's a way to kind of address signals from one cell type to another. And so one thing we want to do is kind of incorporate that kind of an architecture into the system. And in parallel, we've also been using the plant hormone auxin. And we have a paper on bioRxiv, which shows how we can build population control circuits where cells can control their own population sizes. And that paper uses an architecture that Uri Alan and his student Omar Karim discovered, which is a kind of paradoxical architecture where the same signal both stimulates death and proliferation of the cell population, and that gives you robustness. And so we have a paper demonstrating that synthetically, and that's another kind of orthogonal private channel. So I think there's a lot of different ways to add communication to the system.
Thank you very much, Michael, for this amazing talk. We can continue discussion in the forum. All the questions will be posted there, and I encourage the speakers and the participants to continue to interact there. So thanks very much, and we'll move now to our next session.